Thanks very much, and thanks everybody for joining today. So I thought that I would like to talk a little bit about my current postdoc work and some of my PhD work, which is all centered on how we can leverage the diversity in wild populations to learn about disease processes. So just a little bit of background on where I've sort of come from and what I've been up to. I got really geeked on this idea of environmental variation in disease in undergrad. So I've basically been working on that ever since. I did a 2-year fellowship at National Institutes of Health, where I was investigating how changing climate and sea temperatures might change the pathogens that are in marine mammal populations. And then I switched for my PhD to a more experimental approach, where I studied experimental manipulation of nutrition quality and how that influences host parasite dynamics. And my current work as a postdoc with the Soay sheep project of the University of Edinburgh is following up on this idea of environment and host interactions and looking at how the ecosystem within a host, and particularly in a mammalian gut, influences the fitness of an individual. So before I dive in, I also just wanted to acknowledge that all of the work that I'm presenting is the product of a massively collaborative team, whether that's Team Mouse or Team Sheep. The experimental design and field work and lab work, and indeed the analysis, is all a very involved process. So it's definitely thanks to everyone listed here that I am able to speak about what I am today. So for a bit of introduction, parasites are ubiquitous presence across the globe. They occur from everything to, from humans down to plants, and a really key sort of attribute of them is that where they occur in natural populations, they're actually a really important part of the ecosystems where they're found. Despite this, however, the most common way that we study parasites is in a laboratory model, where we have a sort of model species with a very likely laboratory derived strain of the parasite that is typically experimentally infected in the model system and then tracked to see the course of infection. We can learn a lot of really important mechanism from these laboratory systems. However, there is a question of whether they actually translate to more natural populations like humans or wild animals, because they really lack a key context where an individual in a very controlled, sterile laboratory setting is going to be worlds different from an individual that's out there living in the world. And this is something that's being recognized more and more as something we should really pay attention to and take caution when we're translating research from the lab to more natural populations. An example I really like is there's been this Twitter account recently which has basically gone through and it adds the caption "in mice" to studies which are sort of overinflating results from a laboratory system to have public health applications. And it's really, although it's sort of a joke, it is telling us that we should really be careful when we interpret this. Most laboratory studies, understandably, want to minimize variation. So they often use a single, for example, single age, my cursor's working there, a single sex, often a single genotype, and a standardized exposure to the parasite in question. But when we go out into the wild, we really find that there's a huge range of the factors that are controlled in the laboratory. So we have a range of ages, of course, different sexes, genotypic variation. The exposure of an individual to a parasite can vary quite a lot from individual to individual, and also quite key, an individual is exposed to a number of different parasites and commensal bacteria. So what it ends up with inside it is very different from host to host as well. And all these things can play a really important role that we can leverage to understand disease in different contexts. My favorite parasite, and the one that I work on most commonly, are gut helminths. They are parasitic worms that live in the gut of a mammal. The adults in the gut will mate and they produce eggs which are shed into the feces. And then once the species is in the environment, the eggs can develop into infectious larvae, which sort of persist in the environment, and that's where the host can come into contact with them again to become infected. So given that this is a multi-stage process, there's multiple steps along the way here where variation can be introduced that could result in a different number of worms actually being able to sort of take up residence inside the host. What this results in is a really over-dispersed distribution of these parasites in natural populations. So most helminths follow this sort of negative binomial distribution, where we have most individuals on the left here actually have very low burdens of infection. So perhaps less than even 50 worms in their gut and very few. But then we do have a few individuals toward the sort of right tail, which carry very high burdens and a disproportionate representation of the sort of higher burdens in the population. Helminths in natural populations have been studied for quite a long time, trying to tease apart what does drive this variation. So we have a lot of information on what's responsible. We know, for example, that host age and sex and genetics, as well as the condition and immunity of an animal, can drive some of this variation. But importantly, there's also factors extrinsic to the host from the environment that can influence these sort of processes. So depending on the density of hosts, you may have a different amount of infectious material being shed into the environment. The habitat can be important for whether or not the larvae will become infectious and develop. And the diet and resources that are available in the environment will in turn influence the host condition and ability to respond to the parasites. So we already have some complications, but sort of over the last decade, disease ecologists have also been recognizing and appreciating that the host is an ecosystem in and of its own right, and the parasites, of which there are often multiple in the wild, so there's very rarely only one parasite that infects a wild animal. These parasites can have their own interactions, which can influence host health in sort of unexpected ways. And these within host communities include not just parasites, but as I've been more interested in recently, also commensal bacteria that live on the skin, and in particular, those in the gut can really influence sort of the host condition. So we have not only factors from these sort of three ecological scales, which might influence infection, but we also have quite complex interactions that could occur across them. So most of what I do is basically try and design studies either from a statistical standpoint or an experimental approach that'll allow me to try to untangle some of these relationships. So today I thought I'd highlight just a few approaches that I've taken to understanding these processes across scales. And the first one is looking at the environmental context of an individual and how that influences host-parasite relationships. So are there clear drivers of parasitism in a wild population, or is it really just entirely context dependent? I'll talk a little bit about an experimental approach to manipulating the environment of an individual and then monitoring the downstream effects on parasites. And finally, then center in on this sort of complex ecosystem that occurs within a host and how that might feed back up to other scales influencing disease. So most of this work was carried out through my PhD, and my PhD was centered on a wild wood mouse system. The wild wood mouse, or Apodemus sylvaticus, is a really common rodent in the UK, so it can be found in most woodlands across the UK. And their primary parasite is Helminth polygyrus, which I'll just refer to as H. poly from now on. And this parasite can be present in these mice, a prevalence of up to 70%. So it's very common, the mice themselves and the parasite. And to really get at this first question of the relationship between the environment and the host, I used a historical trapping data set from my research group, which included 6 years of data that spanned 6 geographical sites, and included trapping that was carried out over 8 months of the year. And this is a really big portion of a wood mouse's life. These mice only live for about a year, so by trapping 8 months out of the year, we were really capturing quite a bit of their lifetime. It included over 4,000 individuals, which included a range of ages, different sexes, and of course, at every capture, we also knew their reproductive status and body condition. So it really helped us to sort of tease apart these interactions between the two scales. The first step was just trying to quantify what sort of variation we were seeing that might be driven by the environment. So that might be factors like season or year or site. And what we find when we look at sort of these results are just that all 3 environmental factors that we looked at, season, site, and year, had quite a strong impact on how infected individuals were becoming. So on the y-axis here, we have the log of the eggs per gram of feces. So the number of eggs that an individual sheds into their feces can be used as a rough proxy for the number of worms that they have in their gut. That's a fairly common metric for parasites, so that's what we have here. And then on the x-axis, we have the different replicates of the spatiotemporal scales that we were investigating. So we can see on the left that over the course of the seasons, the burdens are really highest in the spring and then decline through to the autumn. This is really common for a lot of parasites. So we often see seasonal dynamics of things like flu, for example, and helminths are no exception. They often have seasonal sort of trajectories. But even across different geographical sites, which were quite close together, and this was all sort of trapped near around Liverpool in the UK, we can see there's quite a variation in the mean intensity across the different woodlands. And over the 6 years sampled as well, we find that some years are much, much lower than others. So the next step was trying to tease apart how this environmental variation might influence the relationship that the host has to its parasites. So just to walk through the framework of what I did for this, I was basically trying to set up interactions between host factors of interest. So for example, here I'll just outline one using body condition as an example. Possibly interacting with the environment. So the environment here might be a year or a site or a season, as we saw in the last slide. So it might be the case in this first plot that there's no relationship between the host factor like body condition and the parasites, but we see the difference in the mean intensity across years, as we just saw. We might also have a scenario where there is a negative relationship between parasites and body condition. So individuals who have better body condition are less parasitized, which is a direction we often expect, but that the magnitude of the effect varies from year to year. Or we could have a more complicated case where not only does the magnitude of effect vary, but actually in different years, we might see that there's a different slope between parasites and body condition. This would be problematic because if we were going to take the sort of global effect here, we might conclude, if we weren't accounting for variation across years, that there's no effect between parasites and body condition. But in fact, what is the true answer is that it's just a dynamic relationship. So if we look at what we find, just as an example from our real data, we see much the same that we have outlined in the figure on the right, where actually the true relationship between the parasites and body condition in these mice looks sort of just like a chaotic pickup sticks. And actually we have some years where the relationship is positive, so 2009 here in green, and 2013 is a slight positive slope, but most years are a slight negative relationship or a null effect. So overall, although we did investigate other factors, I'll just highlight that body condition one here as a sort of example. The answer to this first question is that there are clear but very inconsistent drivers of parasitism in these wild populations. So environment must really be considered alongside the host effects. There could be a number of reasons for this variation that we see across years. So one big thing that I was really interested in was something that varies quite a lot year to year in wild animal populations is the resources that are available to them. And this is really important because nutrition is very vital for all organisms. It is responsible for contributing to factors like growth, immunity, reproduction, general cell maintenance and growth. So we have everything to believe that the diet and resources available to an animal will be really important for their infection and how they respond to the infection and their sort of disease trajectories. There's been a lot of recent interest in this sort of over the past few years because in addition to natural variation that occurs in the environment, so things like climate or just seasonal changes in resources, we're also seeing a massive uptick in the effects of anthropogenic influence on the type and the quantity of food that's available to wild animals. And this can occur through accidental measures like animals being exposed to urban trash, or it can occur even through intentional supplemental feeding like bird feeders or management programs for conservation. So there's a pretty strong motivation to understand how these changing sort of resources and environment will influence the health of wild populations. But this isn't a trivial task because these resources might influence the host via their susceptibility to parasites. So in that case, it might influence their condition in a positive way and make them better able to respond to parasites. But it could also influence their exposure to parasites by creating a sort of aggregation point around the food source, which would then potentially increase transmission. So there's been some recent work, which has been really neat, which has highlighted that across different host parasite populations, we have some wild animals that show increase in parasitism after anthropogenic influence on food sources. and some would show a decrease, which is probably mediated by this condition. But to date, there was no sort of conclusive evidence on how this would play out in a single host population. And this is important because most host populations, as I mentioned earlier, are co-infected with multiple parasites. So really understanding the effects of this nutrition is likely to need the context of the entire parasite community. So my experimental work in this regard was reasonably simple in setup. I went out to wild populations of wood mice that were in Scottish woodlands. I used a really high quality mouse chow, which is a standard feed that you could get for laboratory animals. It's very high fat, high protein. It has lots of micronutrients as well. Added that to the sort of a subset of grids that we designated as supplemented in the wild, and then looked at the downstream effects on parasites. So in each year that I did this, I would set up 4 experimental trapping grids. Half of them was designated as supplemented. Every night I would set out these little traps, which are in this photo here, under a little cover so they don't get wet. They would be baited with lots of food and nice bedding. So it was basically a little hotel mouse. We would check them the next morning and process any mouse that we found, and take body condition measures, samples that would allow us to characterize the parasites, and also their sort of morphometric information, like their size and their reproductive status. So of course, we were very interested in what the effects of this diet supplementation would be on their focal parasite, but this sort of study system is really ideal for getting at this question of how it might affect the broader parasite community as well, because these mice are absolutely riddled with other parasites. So they have not just their primary parasite H. poli, but also a range of other gastrointestinal helminths like Capillaria. They have ectoparasites like mites and fleas and ticks. And they also have bloodborne parasites like trypanosomes, wood mouse herpes virus, Bartonella bacteria, and some protozoans that live in the gut as well, all of which can have impacts on their health. So through our sampling, we were able to perform diagnostics for all of these parasites. For the parasites that live in the gut, we essentially just collect a fecal sample from the trap after every capture. So the mouse will have left behind some poo. We collect it. You can mix it with a salt solution. The parasite eggs then float to the top of that sort of like little setup there. And then you can essentially just put a microscope slide on top and count all of the eggs that are there. So it's a pretty simple but very effective way of enumerating the parasites that are in the gut. For the ectoparasites, we can even more simply brush the fur of the animal when we're in the field and count the parasites that are there. So that poor little guy has a massive tick on his head, but they're often much smaller. And then for the bloodborne parasites, we collect a very small blood sample from the tail and then run diagnostic PCR on these. So this allows us to quantify the effect after supplementation on the sort of parasite, either burden or the prevalence of it, if it was just the presence or absence. And we can compare that to the grids that received no supplementation, which is what I'll show you next. So starting with the gut helminths, again, we have the log of the eggs per gram feces on the y-axis, so a proxy of how many worms are in the gut on the y, and then on the x-axis we have the diet groups. So the supplemented groups here will always be in blue. So we can see for both of the helminths that are present in these mice, the diet supplemented group had much lower burdens, so it seemed that they were better able to fight the parasites off. However, things get a little bit more complicated when we move on to the ectoparasites, because we see that for ticks, the supplementation likewise reduces the number of ticks that the animals have, but we see the opposite effect on mites, where actually the individuals on the supplemented grids have higher levels of mites. Things get more complicated yet when we go to the microparasites. We can see that for both Trypanosoma grossi and Eimeria hungariensis, which is a protozoan that lives in the gut, these both have increased in the supplemented groups. This was a particularly interesting result to us in the case of Eimeria hungariensis, because we know from this system that H. polygyrus, so their main parasite over here, and Eimeria hungariensis, this small gut protozoan, live in exactly the same space in the gut, so they compete for space. So what this is suggesting to us is that the effects of the nutrition on the H. polygyrus lowers the H. polygyrus and then allows the sort of competitive space to open up for Eimeria to proliferate. And this sort of hypothesis is strengthened by the fact that this final parasite, Eimeria uptoni, is another species that is very similar to Eimeria hungariensis, with the exception that it doesn't share space in the gut with H. poli. So it's likely that unless there's that competitive interaction, we just see a reduction in the other Eimeria species. So this indicates that not only are there diverse impacts of nutrition on multiple parasites in sometimes different directions, but that the interactions between the parasites might actually be important as well. And the takeaway here is really that within this single population, we sort of see this range of negative to positive effects. And the implication there is that actually, as summarized nicely by a Viva card that I received from one of my friends, you can have really good intentions sort of supplementing wild populations, but unless you're measuring the entire parasite community, you're not really sure what the health effects are. So the entire community of parasites is really key context for being able to predict what the effects of resource provisioning and changes to the environment for an animal might be to wildlife health. And it was this sort of within host complexity and the fact that these interactions might be having such key effects on the parasites that was one of the things I was really keen to follow up with on my postdoc work. So that really led me to the work that I'm doing right now, which is considering not only the within host populations of parasites, but also their commensal bacteria in the gut, to look at what determines the sort of structure of those communities that live inside the host, and also what the consequences of those communities are for the host fitness down the line. So this has been work that I've been involved in since this September. It centers on the fact that individuals are host to these incredibly complex communities, so both parasites and sort of bacteria. There are trillions of bacteria species that can infect a mammalian, that can be present in a mammalian gut. And increasingly, there's building evidence that the gut microbiota in particular can have really important effects on host immunity, host metabolism, even on sort of mental performance in some cases. And this is being sort of recognized more and more, and it's starting to show up in the way that people design experiments. So there was a really cool study in 2019 where the authors essentially took embryos from a laboratory mouse, and they put that embryo into a wild mouse, which had had a natural microbiota, had been exposed to pathogens in a natural environment. And then they looked at the offspring, which they called wildlings. And what they found was that these wildlings had immune responses that were much more similar to a human or a wild animal compared to laboratory mice, which often fail to replicate things like clinical trial results for humans. So this is a pretty important step between this laboratory to wild spectrum, and indicates that we can sort of start to tease apart why it might be that some of the studies that happen in the lab fail to translate to natural populations. This appreciation of the sort of natural microbiome and why it might be important for individuals has been part of what's led to a huge expansion and explosion of wild microbiome samples. So over 2006 to 2019, you can see on the plot on the right here that the number of studies which are sort of aiming to quantify the gut microbiota in the wild has just rapidly climbed. So now being in this field, I see new papers just almost every day that's sort of quantifying the gut parasite communities. But it's a really tricky thing to study, because you can imagine these complex communities exist within an animal that already exists in a very complex environment. So the work that I'm involved with now is aiming to take a really multi-step approach that will allow us to sort of systematically unpick the complexity of these relationships. And this is in collaboration with the Soay sheep project in Edinburgh, which is a really long-running, long-term wild animal study. So the first step here is really just trying to determine what are the host and environment factors that drive the communities that we see in the gut, so illustrated here on top of the sheep. The second step is then to tease apart how the bacteria are interacting with the parasites and interacting with gut protozoans like Eimeria that I mentioned earlier. And then with an eventual aim to try and apply this to the fitness data we have of these sheep to really understand how these within host communities are influencing the disease that we see in the population. This is a really great system for this, because we have over 10,000 sheep on the island on St. Kilda, which have been studied since 1985 continuously. So we know the relationship between all the sheep, we know who's mother is who's had this so many children. We have extensive life history and phenotype information and immunology information for them. Even more recently, there's been more detailed genetic information for them. So these sort of SNP chips give us an even more sort of in-depth view than the pedigree. And this new grant that we're working on is expanding the efforts even more. So we have in-depth wild immunology going on. We're using next generation sequencing to identify the community of parasites within the gut, and then population level modeling as well to really link these factors across scales. The basics of the approach, and most approaches for microbiome studies, are we're essentially collecting a lot of fecal samples from the individuals. So a very common thread of my work is that it's fecal sample based. We then extract the DNA from these samples, and then they have next generation sequencing techniques applied, which give us then a massive library output of reads for just lots of different bacterial species. The most common way that these are dealt with is that after they are sequenced, they are generally trimmed and processed for quality control. And then sort of after the second dashed line here, this is just depicting that they are then often grouped into what are called operational taxonomic units, or OTUs, based on a certain threshold of similarity, so something like 97%, and then transformed into, within a sample, every OTU having a certain proportion of that sample. Another technique is to randomly subsample from the number of reads that you get back across each sample, to try and standardize across different library sizes that you get for different samples. And that's based on the assumption that you end up hitting a plateau with your species discovery at a certain point. We're taking a slightly different approach because both of these approaches that I've outlined here can have limitations, mainly that it's not actually using the entirety of the information that we have. So we have, in the data that we get back, raw abundances for every sequence variant that occurs. So rather than grouping things to an OTU level, we're first of all using the exact sequence variants, and we're second of all using the raw abundances for each of those present in every sample to model. So we have this pilot data set from 2013, where we have nearly 60 individuals from a group of lambs and adults. So we can look at whether or not, as a starting question, these communities shift as animals age. So this includes 5,000 exact sequence variants that we get back from the sequencing data, and it results in 270,000 reads. So that's the sort of row number of our data set that we're working with. And working with sort of one of my supervisors, Luke McNally, who developed this mixed model approach for teasing this apart, which looks a bit scary, but the basics of it are that it's using these sort of raw abundances as the response, which details the number of reads for a sequence variant in any given host, and then adding in random effects in a mixed model framework, which allow us to, in the purple box, control for factors that might influence abundance, and in the blue box, allow us to really tease apart the factors of biological interest that we care about. So here, the interaction between the taxonomy or the sequence variant and age would tell us whether or not the community is shifting. So that's really all you need to worry about, not really the equations, just that it allows us to break down factors in the environment and host level that are explaining variation in the microbiota data. So that's the main output of these models that I look at when they've run, is how much the factors that I care about are explaining the variance in the model. So here's an example of one of the model outputs. What we can see is that across the terms that we have in the model, so that includes a term that explains just the distribution of the data itself, which follows a Poisson distribution. We have a term which just indicates the taxonomy. So this is just telling us that the abundance is very variable across the different bacterial taxa, which makes sense. We have a very small effective sample, so there's no, the read depth is quite good across the samples, that's telling us. But what we care about here is really in orange, indicates that the effective age on the composition explains 17% of the variation in the data, which is quite a lot, considering that most of these types of data sets have a lot of residual variance, as we can see below here in yellow. So this basically indicates that the individuals in this photo are very likely to have very different community compositions. And I'll highlight very quickly, just to end, that the sort of next steps where we're taking this is really trying to then tease apart, okay, so we know that age influences the community composition, but can we actually see which bacterial taxa are responsible for these effects? So what I can do is extract information on the models that allow me to look at the shifts in the specific bacteria taxa. So the plot that you'll see next is showing the effect of every individual ASV, every individual sequence variant, and whether that sequence variant shifts so into adulthood more or less. So if the point is above 0, then that indicates that the sequence variant is more common in adults, and if it's below, it's more common in lambs. The bigger point is more confidence in our estimates. That's really all. And what we can see here, if we look across all of the phyla that we find in our study population, that the host effects of age are really variable across taxa. So a lot of them cluster around 0, is what the violin plots are showing. But a few of them, so in particular the Bacteroidetes, we see that they sort of have a tail in the positive direction, indicating that this taxa is much more common in adults. The Firmicutes, on the other hand, we see the sort of tail in the negative region, indicating that that's more dominant in the lambs. And we can pull out these top 50 and bottom 50 effects, and that backs up essentially what we can see sort of anecdotally by eye, that the age effects are really being dominated here by these two classes, the Firmicutes and the Bacteroidetes. And these are two phyla which we know are really associated with aging, in particular in humans, and also with metabolism. So we're starting to get a sense here that being able to pull out these sort of taxonomy specific results might start to give us an indication of what the functional changes are across ages, or indeed across other host factors that we can build into the models at a later point. So just for a couple take-home messages to reiterate, we find that understanding the drivers of the disease in the wild really does require accounting for environment to understand the host-parasite relationships, that the context of the within host community of all parasites is probably very important for understanding environmental effects on health. And we're starting to see with the work I've been involved with recently that within host communities can in fact change according to age and likely other host or environment factors, and will probably have important consequences for immunity. So the future work that I'll be sort of engaging with this postdoc is then trying to like further strengthen the links we have across these ecological scales. And that's me.